Thank you, Dr. Hardik. I think the first and the third talk actually are interlinked. I don't know why I got sandwiched between the first and the third talk. This will have nothing to do with inflammation or anything. I'll basically be talking about positioning of a biotech innovation that has actually come from India, and it's a voyage from the clinical trials to the real world evidence. Now, whenever there is a biosimilar, people look at it with a little suspicion. They start asking questions that, okay, is it very similar to the originator that we already had and we had confidence on? In fact, it's very difficult to make a biosimilar because it's not like any other process, because especially if you're doing insulin, it's extremely difficult to make a biosimilar insulin. Okay, I can just give a very, very odd example that if you have Priyanka Chopra, who is an actress, and then if you want to make another Priyanka Chopra, it becomes that much more difficult. I mean, you don't get an exactly same Priyanka Chopra, but you get a similar Priyanka Chopra. So the processes that are involved in making a biosimilar are very, very stringent. Now, what do we look for in a biosimilar insulin, and what is it that the authorities who approve it look for? They are very strict and rigid about it. Now, I've just taken the example of three big names: the EMA, which takes care of the European sanctions, the FDA, which takes care of the US, and PMDA is from Japan. They have slightly different set of rules. Till now, the DCGI did not have the rules, but now even the Drug Controller Government of India has now set rules for biosimilars. Now, you need PK/PD studies, and with EMA, you need a phase 3 study in type 1 to run for 6 months. FDA also needs PK/PD. Now, that's common. They need a phase 3 study in type 1 for 12 months, but they look at the primary efficacy parameters at 6 months. Now, very similar in the PMDA for Japan. Now, if your product is there apart from India in either US or Europe or in Japan, gets approved in the other countries, they have to go through very rigid and very strict parameters to satisfy that everything is in place. To you and me as clinicians, a very simple definition of pharmacokinetics tells us about the absorption, distribution, metabolism, and excretion, and pharmacodynamics tells us the relationship between the drug concentration and the site of action and the resulting effect. It also tells us about the time course, the therapeutic, and the adverse effects. So this has to be very similar to the originator when you are actually doing the PK/PD studies. So I'll present you the PK/PD data done on type 1 patients. This is from insulin glargine that was made by the Biocon. Now, the objectives are to look for PK/PD bioequivalence of the Biocon glargine with two references. One is the US reference glargine and a European reference glargine. Now, the reference glargine always is the gold standard, and then we know in this case it is the Sanofi's glargine which was compared. Now, there can be batch-to-batch variation. I will not go into the process of making insulin. So there can be batch-to-batch variations. So when you do a PK/PD study, you compare it with the European reference and you also compare it with the US reference. And to that extent, you have to show, okay, my product is equivalent to both these products. And this was done on type 1, 114 patients, a single center which gets accepted world over as a center of excellence when you actually give a data. I mean, you can't just do PK/PD studies in some small lab and say that, okay, I did it. Now, it has to be done in an approved center, which is approved by the FDA, PMDA, and the EMA. Now this is the crossover design. I will not go into the details, but I'll just show you the curves. Literally, the insulin glargine from Biocon compared to the US glargine and the European glargine. If you look at this curve, they're literally hugging each other. And you come to know which is what only if there is a color code. Now, if you remove the color code, you just can't even make out what it is. Exactly similar to the US and the European reference glargine. Now, when you look at the pharmacodynamics, very similar curve, literally hugging each other. So there's absolutely no difference between the US reference glargine and the European reference glargine when the Biocon's glargine went through PK/PD studies. So this is the conclusion, what we just spoke about. They're extremely similar, very well tolerated, and there's no significant safety issues at all. Now this is the global phase 3 study to be done in type 1 patients. Remember, insulin is something that we use in both type 1 and type 2. Now, it's extremely important to show that your insulin is very similar to the gold standard insulin, both in type 1 and type 2. So, the regulatory market demands that you do these studies before you come for approval of your things. So, the InStride 1 was done on, it was a phase 3 trial done on type 1 patients. Look at the number of people required here, 558. Now you should remember, now for biosimilars, you don't need large numbers. You don't need 5,000, 6,000, 7,000. The number is decided by the protocol and the power that this number can give. And then in this case, it was 558. Now this was the study design. When for 52 weeks, the primary endpoint was a change in HbA1c from baseline to week 24, and then to prove the non-inferiority margin, it should not be greater than 0.4% at week 24. So that's the confidence interval at 95%. You also have secondary endpoints, HbA1c change from initial administration to week 52, change in fasting glucose, change in insulin dosage, and the SMBG frequencies. Then there were safety parameters like hypoglycemia or any adverse events, and they also looked for immunogenicity. They were looking for anti-drug antibodies because when you are using a new insulin, it should not be the anti-drug antibodies. Now this is the HbA1c over time, absolutely no difference between the biosimilar glargine versus the standard. And when it came to the non-inferiority margin, it was absolutely less than 0.4%. Now that's the conclusion there. Now look at the actual glargine dose over time between Biocon's glargine versus the gold standard glargine. No difference at all, exactly the same. Nocturnal hypoglycemia, both exactly the same, so there was no difference at all. So the conclusions are very clear. The biosimilar glargine of Biocon is very similar to the gold standard that was used here, and it proves non-inferiority of insulin glargine. Changes in HbA1c in fasting were similar. Both basal and bolus insulin doses did not differ from two groups. Rate of hypoglycemia or other AEs were comparable between two groups. Now this is a phase 3 study in type 2 diabetes. A very similar design, 560 people. This was a randomized trial, 1:1 randomization, very similar study endpoints. Similar secondary endpoints. Now the actual HbA1c over time, very, very similar. There is no difference in two groups. Non-inferiority again proved by the biosimilar insulin. The actual glargine dosage over time remained exactly the same in the two groups. Hypoglycemic episodes very similar in the two groups. So again in type 2 patients, like what we saw in type 1, there was no difference between the biosimilar insulin of Biocon versus the gold standard insulin from Sanofi, in this case of course the Lantus. Now look at the immunogenicity in these two studies, because that's important to know that there's going to be no cross-reactive ADA, that is the anti-drug antibodies, both in stride 1 in type 1 patient and in stride 2 in type 2 patients. None of these autoantibodies were found. Similar graph as a bar chart, it tries to tell us that in type 1 you can expect a little more antibodies, but then in this particular trials, it was not to be seen. It was very similar to the gold standard insulin. So this is the conclusion from the immunogenicity assessment. We do see in type 1, but it was very similar in both the set of patients, both in the biosimilar insulin versus the gold standard. So what are the key messages of these global clinical trials? The PK/PD properties of Biocon's insulin glargine and the reference insulin glargine from both US and EU were determined as equivalent. In stride 1, both insulins did extremely well. In stride 2, again, insulin was very effective and had similar metabolic and safety profiles. When you look at the immunogenicity profile, it was comparable immunogenicity profile between the two groups in both in stride 1 and in stride 2 trials. Let's look at some real-world evidence from India itself. It's still ongoing actually, multicentric, covering 816 cities and towns in India. This was basically to assess the efficacy of the Biocons, both human insulin and insulin glargine. Okay, there's no PK/PD. Efficacy-wise, they looked at the fasting blood glucose values and the change over time. Patients are doing extremely well. Postprandial over 9 months, patients are doing well. HbA1c over time. So this is basically an efficacy study which they needed to be done for the DCGI purposes. So it proves that the fasting postprandial and the HbA1c responses are clearly seen in the Biocon's insulin. So 2018, this is the big news. In 2009, the glargine was approved in India by the DCGI. Then 2015, they got the approval in Mexico. 2016, the approval in Malaysia. 2016, again, it was the first glargine from India to get a global approval by the PMDA of Japan. 2017, global data unveiled at the ADA. 2018 now approved in South Korea. So it's available in 28 countries and the TGA in Australia. Now it's an approved biosimilar glargine made in India for the globe. Not yet available in the US. It's just a question of time before it's going to be available in the US. So thank you very much for a patient listening. I finished 7 minutes earlier, so thank you very much. Next speaker gets that much more time and we have some time for the question and answers.
But since it's a different session, so we can have question answers for this lecture. If anyone wants to ask anything. No, but yours is a different topic, so.
Topic, that's what he says. Okay, the first and the third talk are very similar, interlinked. So if there are questions, I can take it now or I can come back at the end.
So I had a simple doubt that they have perfect efficacy-wise and effect-wise. What about the antigenicity?
Antigenicity, I just showed the data both in stride 1 and in stride 2. They call it the ADA, anti-drug antibodies. It was very similar to the reference product. In this case, the gold standard was the Lantus that was used, both in type 1 and type 2. So there was no immunogenicity problems with the biosimilar, because if there is any problem, it's not going to be accepted in either EU or in Japan. So the very fact that one of your product gets approved in either EU or in Japan or US, that very clearly says that it has gone through very stringent safety parameters. So there is no problem of immunogenicity.
We have Indian data towards that.
Sorry?
Indian data.
Indian data?
Yeah, towards the antigenicity in our country.
Indian data towards antigenicity is, you have the data?
Because the DCGI submission required the data.
Okay.
It initially didn't require, but they did anyway the immunogenicity study.
Okay,
it is
so
there.
I had some real world data on
part of the study, sir.
which I had given it to them, but there is not a published data. So I said don't use it, but if it has been presented to DCGI, I'm sure we can ask for it. It must have been presented to DCGI for approval.
Thank you, sir.
What about safety in pregnancy?
Well, glargine is not approved anywhere for pregnancy. I mean, your FDA does not approve glargine to be used in pregnancy, but the Endocrine Society in US have approved the use of glargine. If you are using glargine in pregnancy, it's off the record, it's off the label indication. So ideally, you have to take the consent of a patient that you are using a medication which has not been approved. Okay, now this is something very tricky. 90% of us actually use Lantus in pregnancy and nothing has happened. ACE have said, okay, you go ahead and use that. Only Levemir has been approved as a long-acting NPH, and Levemir has been approved by FDA. Glargine is not approved, and now you cannot do any clinical trials in pregnant patient anyway. So I don't think anybody can get data of what exactly happens if you use it in a pregnant lady, because you're not allowed to do a trial.
Thank you.
Do we have any other questions from the audience? No, I think you made it
Thank you.
clear. Thank you, sir.